Kind of series of talks this morning focus on biological and neuropsychological studies of drinking and drug use in college populations. I'm really pleased to introduce our first speaker in this session, Dr. Krista Medina. Krista completed her doctorate with an emphasis in clinical neuropsychology at the University of Cincinnati, and then completed a NIDA-funded postdoc at University of California at San Diego, where she focused on adolescent brain development, addiction, and neuroimaging work. She's currently an assistant professor of psychology at University of Cincinnati, where she serves as the director of the Brain Imaging and Neuropsychology Laboratory. Dr. Medina has published over 20 articles that help characterize the long-term effects of drug and alcohol use on the developing brain, focusing primarily on adolescents and young adults. And her research also attempts to explain individual differences in the drug effects on the developing brain. Her research conducted at University of Cincinnati is supported both by the Center for Environmental Genetics and by the National Institute on Drug Abuse. And the title of her talk is The Effects of Drug and Alcohol Use on Neurocognition During Emerging Adulthood.
Sure. That'll work. Yeah, thank you. Good morning. I don't really have any excuse. I am on East Coast time, but I'm chronically tired because I have a 2-year-old, so that's my excuse. I need to warm up here. Does this thing work? Okay. So today I'm going to talk about why I got particularly interested in studying the effects of chronic drug use on the adolescent and kind of emerging adult brain or young adult brain. And then I'm going to share some of our findings on the effects of binge drinking on the teenage brain as well as the young adult brain, and then go into some of our findings on the effects of chronic marijuana use, and then talk about some potential limitations of this research and then also future directions. So I'm sure as most of you would agree, addiction really has a teenage age of onset. And so the vast majority of individuals start using drugs in their teenage years. And really the introduction of substance use disorders peaks in the emerging adulthood or young adulthood years. Although most of our studies really looking at the chronic effects of drug use is actually in older adults, especially men, especially vets. And so I was really dedicated to trying to look at the effects in the younger brain, really when the drug use is introduced. And of course, we've looked a little bit about the epidemiology of drug use, but this is just another slide showing you that even in 12th graders, a pretty large percentage of our teens in the past month have gotten drunk, almost 30%. And about just over 20% are using cigarettes, and just short of 20% are using marijuana. About 6 to 9% are using marijuana daily. And this, again, is showing that binge drinking is very prevalent in young adults. You can see it's even worse in those who are enrolled in college, which is the top line there, just over 40%. And that has actually been fairly consistent since the 80s. So this has been a problem, indeed, that we've been dealing with for a while. So not only is there an introduction of drug use, but we have some very dynamic changes in the brain during adolescence and even going into the early 30s, which is good news for some of us. After that, we don't really need to talk about what's going on. So basically, we have changes in the gray matter. So we have this pruning process where there's actual cortical thinning, especially in the prefrontal cortex. And that actually occurs up to age 25, which I don't know why we needed this research, because the car rental places have been telling us for years that the risk for driving goes down at 25, right? But the neuroscience is catching up with Hertz, and we can see that at around 25, the prefrontal cortex is finally looking more adult-like in at least its gray matter volumes. White matter development continues up until the 30s. The other thing I want to say is there's substantial differences in development between boys and girls. As we all know, girls mature earlier, And now we have the neuroscience to back it up. So the girls' kind of peak brain development occurs about 1.5 years earlier than boys. And so there really may be very different trajectories and the effects of drug between men and women, especially during adolescence, young adulthood, before the brain is finished developing. The other thing I want to mention is that the prefrontal cortex comes online after the limbic system. And so the limbic system, including the amygdala and the hippocampus, is really related to emotional regulation, stress response, things like that. And there's some wonderful animal research showing that stress exposure in the teen years lasts much longer and is experienced much worse both in humans and animals. And so the experience of stress and emotional problems in these adolescents and young adults really is very qualitatively different than what's experienced in adult years. And so with that said, let's talk a little bit about the effects of binge drinking. So we do have some research showing that more problematic alcohol use in alcohol use disorders, so abuse and dependence, is associated with several different brain changes in teenagers and some young adults. For example, we found in teenagers with alcohol use disorders, we see poor cognition, especially in memory, attention, and visual spatial skills. We also see reduced left hippocampal volumes. And in girls, we see reduced prefrontal cortex volumes, as well as abnormal brain activation on fMRI tasks. And so there is some evidence so far accumulating that alcohol use disorders in teens is associated with abnormalities in brain function. But what we've been asking recently is, what about binge drinking? So what about before they even really develop alcohol use disorders, but just some of this more modal binge drinking that we see in the 40% of 18 to 25-year-olds. And in this series of studies, we've kind of defined binge drinking. This is some of Susan Tapert's work at UCSD, as well as my work at the University of Cincinnati. And across the studies, we've defined binging as 4 drinks for women and 5 drinks for men in one episode. In Dr. Tapert's research, they're looking at binging in the last 3 months. I looked at binging over the past year, doing a year-long timeline follow-back, so actually looking very closely at the patterns of use. We do have information on kind of peak binging within 2 hours, but the metrics during the past year, it's during kind of one sitting instead of the 2-hour limit. And this is a study that is by a graduate student of mine that came out fairly recently. They looked at 14 binge drinkers and 14 matched non-drinkers. And this is the teenage sample, about 16 to 19-year-olds. On average, they're about 18 years old. So they're mostly high school seniors and freshmen. And what they actually looked at is diffusion tensor imaging, which is actually an imaging technique that looks at quality of white matter. So it's looking at, if you can imagine, spaghetti noodles that are uncooked and you put water through, not much water gets through because it's very tightly packed. But if you look at cooked spaghetti and hold it, a lot of water is going to get through. And that kind of represents poor integrity of the white matter tracks. And what they found in these 14 binge drinkers is they had reduced white matter integrity or increased water diffusivity through white matter tracks in about 18 clusters throughout the brain. So it was fairly diffuse. And it was particularly concentrated in prefrontal cortex regions and a region that leads to the hippocampus. And as you can see here, in about 6 clusters, you see a dose-dependent relationship with hangover symptoms, which is really a marker of how bad did the binge get, if you can think about it that way, and really reduced white matter integrity with withdrawal symptoms. And I really want to emphasize that these are healthy teenagers without comorbid axis 1 disorders. Okay, so they don't have conduct disorder, they don't have ADHD, they don't have mood disorders, they have relatively low depressive symptoms, they have very low other drug use, a tiny bit of marijuana use in there, and they do not really meet a criteria for alcohol use disorders. So this is just binge drinking. With that same sample, slightly lower because they didn't have usable fMRI data on a couple of the teens, Alicia Schweinsberg, who's now at Yale, but who worked in Dr. Tapert's study, found that those binge drinkers also had poor verbal encoding. This was a marginal difference on an fMRI task. but they didn't learn new words as efficiently as non-bingers. On top of that, they found some abnormal fMRI response, increased prefrontal cortex, cingulate and parietal response, with decreased occipital, precuneus, and parahippocampal gyrus activation. So they had some really abnormal brain activation that shows perhaps some compensation with prefrontal cortex and parietal regions. They looked specifically at the hippocampus because this is a verbal encoding task. And what they found was very actually subtle differences. And so they probably need a much larger sample to start tapping into hippocampal differences. But basically, the controls engaged the hippocampus significantly more for this encoding of new material compared to old material, while the binge drinkers really didn't show that pattern. And so there were some subtle differences in hippocampal activation. And this is some of my data that isn't published yet, so I want to orient you a little bit more to the sample since it's not out there. I have 18 controls and 21 bingers, and they underwent a neuropsychological battery. For the purpose of this talk, I'm actually focusing on verbal memory function and hippocampal volumes. And I chose to focus on those first because there's some great animal research showing that binge drinking seems to be particularly neurotoxic to the hippocampus. And so if there's going to be a biomarker of problematic impact of binge drinking, it's probably going to be in the hippocampus. Our sample has about, on average, 2 weeks of abstinence, although it kind of ranged from 3 days to a few weeks. And the bingers were defined as folks who had binged at least twice in the last 3 months and greater than 6 times in the past year. Perhaps more importantly was the controls, because frankly it was hard to find controls who didn't have too much binge drinking, because this was more of the college population. They had 0 binges in the last 3 months, and they had less than 4 in the past year. I think I had maybe 10 people who had never binged in the past year. So there's a tiny bit of binging even in the controls, but I actually looked at a dose-dependent relationship. They did not meet criteria for an alcohol use disorder, and they couldn't be defined as heavy drinkers using Cahalan criteria. I will say about 30% of the bingers did meet criteria for an alcohol use disorder. Majority met criteria for abuse. Other exclusionary criteria includes independent axis 1 psychiatric disorders. Now, there are people in the study who met criteria for a major depressive episode, but it was deemed to be substance-induced. They weren't on any current or past psychiatric medications. They didn't have a family history of bipolar or psychotic disorders. They couldn't have any neurologic or major medical illnesses, and they couldn't have significant illicit drug use except for marijuana, which is, you can't separate marijuana from the heavier drinking. So the controls and bingers, this is just to kind of show that they're well matched in age, education. I've tried very hard to balance them for males and females, so they are 50-50 male-female. Majority Caucasian, balanced in their reading ability and their depressive symptoms, and as you can see, they're below clinical thresholds for any current depressive symptoms. They are, now I'm focusing on binging, but they are significantly different really on just about every indice of alcohol use. For example, the age of alcohol use onset, so regular alcohol use, the bingers did start drinking earlier. They had about 37 binges on average in the past year. Their alcohol drinks in the past year, the bingers had about 290 standard drinks in the past year compared to the controls who drank about 20 drinks. There were no heavy drinkers or folks with alcohol use disorders in the controls, whereas about 62% of the bingers were considered heavy drinkers and about 30% had alcohol use disorder. There was some marijuana use in the bingers as well as cigarette use in both groups, and that was statistically controlled for any comparisons. And what I find in multiple regressions and controlling for things like ethnicity, gender, income, reading ability, depressive inventory symptoms, cotinine levels, which is really a marker for how recently you smoked cigarettes, and past year marijuana use, I found very robust relationships actually between the number of binges in the past year and poorer verbal memory. And this was in delayed recall, retention of material that you had already learned. So those words that you learned by the trial 5 or the 5th exposure to words, how many of those did you remember after 20 minutes? And recognition of information. And I have to say it's rare to see recognition deficits, especially in folks who have only been drinking for a few years and don't have alcohol dependence. There are significant binge by gender interactions, and I'll show you what those meant. The bottom line was, at least in this sample, the results were really driven by the men. The women didn't have a very strong relationship between binge drinking and verbal memory, whereas the men had a very robust negative relationship. I then looked at hippocampal volumes. So we hand traced left and right hippocampal volumes in this sample. And we found actually for both the men and women that increased binging in the past year was related to smaller left hippocampal volumes. And this kind of green line is showing you the average. And so basically, once people started binging more than about 20 times per year, their hippocampal volumes were below average. And this is the men and women. So even though we didn't find a relationship in verbal memory for the women, we do see it in the men. And that may be because women's verbal memory tends to be superior. And so it may be that it's a little early in the disease process for us to be measuring on a neuropsychological test any functional impairment in the women. On top of that, we actually see abnormal brain behavior relationships. So this is to just confirm that smaller volume was bad, because in teenagers and young adults, there's this pruning process, and so sometimes smaller volumes is actually a good thing. And that's true for the controls, actually, which is this blue line. So smaller volumes, I'm sorry, am I screwing that up? The blue line, yes. Anyway, smaller volumes in the binge drinkers was associated with poor verbal memory. So small volumes was a very bad thing in this sample. So in summary, we found that increased binge drinking in teenagers was associated with reduced white matter integrity, especially in prefrontal cortex and hippocampal regions, and reduced parahippocampal and increased frontal parietal brain response to a verbal encoding task. And then in our young adults who are slightly older and who had been binging a little bit more than the teenagers, we do find even deficits on neuropsychological tests, including delayed recall, retention, and recognition of verbal memory. And this was correlated with significantly reduced left hippocampal volumes. So let me go ahead and move on to some of our findings related to what happens with chronic marijuana exposure. And so this was one of my first studies looking at marijuana effects. And this is 31 teenagers who were chronic marijuana users compared to about 34 controls. And I will say this is actually after a month of monitored abstinence. So they come into the lab and they, about every 3 to 4 days, and get urine tox screens for a month to show that they're getting marijuana out of their system. And after controlling for comorbid alcohol use and any demographics, we find that increased marijuana use was significantly associated with poor sequencing ability, psychomotor speed, complex attention, especially sustained attention, and story learning. And the whole picture really was related to that marijuana use seemed to be impacting them, that they're thinking a little slower, and once we really made things difficult, they couldn't stay online. But they could do simple attention tasks. These teenage marijuana users also had lower grades and more problems in school, and so there did seem to be some functional impact of their cognitive status. I went on to do a couple of structural studies, really focusing on the prefrontal cortex, hippocampus, and cerebellum, which have a lot of cannabinoid receptors in them. And I find that in the women, actually, we have increased volumes in the prefrontal cortex compared to controls. And again, bigger was not better in this case, so larger volumes were associated with poor executive functioning. We saw kind of similar effects actually in the cerebellum. So this is a region in the cerebellum vermis that actually has high connections with the limbic system and the prefrontal cortex. And we found, again, larger volumes in the cerebellum in both the men and women. And this was related to poor executive functioning. And so we also found in another study increased left hippocampal volumes associated with marijuana. And so taken together, we think that perhaps chronic marijuana use actually may disrupt this healthy pruning process. And so if you're not getting rid of poor synaptic connections in your brain, you're going to have a larger volume. And so they almost look like they have more immature brains. And they have poor white matter integrity, which is going to make you less efficient. So if you don't have good white matter connections, and you have all these synaptic connections that are actually fairly weak, you're going to be slower at processing, and it's going to affect things like psychomotor speed and sustained attention, which is exactly what we see in our neuropsychological testing. And this is some of my new data that really confirms some of these findings in young adults. So this is about 18 to 26 college population. Again, well matched in the important demographics, although I will point out that the marijuana users are approaching significance in having larger body mass index. Their weights are significantly larger. So a lot of, I don't know, munchies. Again, they do have more alcohol drinking, which you just, if anyone has any advice on where to find marijuana users who don't drink, let me know. I thought California would really be the best bet, but even there. They have a smattering, a tiny bit of other illicit drug use, but really very little. And what I found is that in this sample, increased marijuana use was associated, again, with poorer sequencing ability, which is the trail-making test, poor sustained and selective attention, and poor cognitive inhibition. And we have some kind of interactions with gender. And this isn't the topic of this conference, but I also want to point out that body mass index was very robustly related to poor cognitive inhibition. And this is controlling for gender and reading ability, all the hand waving, all the other stuff. And this is just, again, showing that a dose-dependent relationship between increased joints smoked in the past year and decreased psychomotor speed and sequencing ability. And again, the results were really driven by the males. And we see, again, that especially in the males, we have decreased sustained attention. And I really just, again, kind of wanted to point out some of the findings with BMI, that increased BMI was associated with poor cognitive inhibition. And this is true for both groups, but particularly true for the controls. And the same thing with cognitive inhibition. So this is a Stroop task, if you guys are familiar with that. Increased marijuana use, poor cognitive inhibition, and also true for increased BMI. And so I think the BMI issue may be a point of intervention that we could do to improve some of the executive functioning. So in summary, increased marijuana use was associated with poor neurocognition. In teenagers that were between the ages of 16 to 19, we saw reduced story memory. We didn't see reduced memory in the young adults, so that may be something that is a little more sensitive in the teenage years. But we do see reduced psychomotor speed, complex attention, and cognitive inhibition in both samples. And Dr. Tapert's sample is primarily males, and so it may be that these results are primarily driven by the males across both samples. However, we do see some structural differences, both in the men and women. Increased prefrontal cortex volumes in the girls, increased left hippocampal volumes and vermis volumes, and in the case of the vermis and the prefrontal cortex, increased volumes, poor executive functioning. So there are some potential limitations. Right now, this research is really at the cross-sectional level. Dr. Tapert's cohort is being followed longitudinally, and we're able to, starting to analyze some of that data. But it may be possible that pre-existing differences explain some of these findings. So pre-existing executive functioning deficits may put you at risk for using drugs, especially cognitive inhibition. On top of that, there could be subclinical conduct or attentional symptoms within these samples, even though we exclude for Axis I disorders. Although I will say that the mood symptoms were controlled for in all analyses. But we really do need longitudinal studies of at-risk adolescents before drug use is introduced, as well as longitudinal studies following treatment or natural abstinence in these users to see if there's recovery of function. And we really need more studies with women and girls in it. Some future directions for my work is to look beyond some of these cognitive components, but also to look at things like stress regulation. You know, how does chronic alcohol use and chronic marijuana use affect the brain's response to stress and hormone response to stress? And how do these relate to treatment outcomes? How do some of these neurocognitive variables affect treatments? And could we use some of this neurocognitive information for prevention campaigns? We know in adults, by giving some feedback on some of their effects of their chronic drug use on their brain structure and cognition can help treatment outcomes, but we don't know as much with adolescents and young adults. We also don't know if there's certain people who are at particularly high risk based on their genotype or comorbid disorders. And we don't have too many interventions to change the cognitive effects. And so if you can imagine, if you're a chronic marijuana user, is there an intervention that we can do to help improve your sustained attention and your processing speed so that you can get back engaged in your studies? Some potential areas would be cognitive behavioral therapy. There are some brain imaging studies showing that doing very short CBT interventions can engage the prefrontal cortex and kind of reduce the limbic response to improve emotional regulation, or executive functioning or working memory training that engages the prefrontal cortex. There may be some medications that help. And I do wonder if reducing body mass index and exercise interventions may particularly help executive functioning as well as hippocampal functioning and structure. And very quickly, I just wanted to acknowledge the funding agencies, especially the National Center on Drug Abuse, some grants to Dr. Tabert and myself and NIAAA, the colleagues out at UCSD, including especially Susan Tabert and Bonnie Nagel, and my colleagues at the UC Brain Imaging and Neuropsychology Lab. And thank you.